So it's a great pleasure to introduce Woj Wojtowicz as our CTSI speaker today. Woj came to UCLA as a graduate student with an interest in immunology and virology, but became captivated by the question of how neurons discriminate between one another during neural circuit development. She joined my lab shortly after we discovered that RNA splicing at the Dscam 1 locus generated some 38,000 different isoforms of an Ig superfamily protein, each with the same domain structure but differing in one or more of three variable Ig domains. And during her first year in the lab, she discovered that different isoforms have discrete binding specificities. She then went on to test thousands of pairwise combinations for interactions, define the binding rules, and with Mike Sawai and David Eisenberg's lab, uncovered the structural basis for this specificity. These beautiful biochemical discoveries inform genetic studies, demonstrating that this extraordinary biochemical specificity acts in vivo for neurons to discriminate between self and non-self, thereby establishing the molecular basis of self-avoidance. During Woj's graduate study, she developed a powerful assay that allows for high throughput screening for interactions between cell surface proteins, and envisioned that this would provide a way to explore the extracellular interactome, much as one uses the yeast-2 hybrid screen to explore interactions between intracellular proteins. As an independent Bose Fellow at Berkeley, Woj set out to identify regulators of synaptic layer specificity in the mammalian retina. Here she used the high throughput interactome to characterize interactions between cell surface proteins expressed in the mouse retina. Through these studies, she discovered some 50 previously unknown receptor ligand pairs. This included two protein families that she went on to show exhibit repulsive signaling in vitro and contribute in this way to layer-specific arborization. Woj then went on to lead a collaborative effort between Chris Garcia, Kai Zin, and others at Caltech and Stanford to utilize the approaches she had developed as a student and postdoc to carry out a massive screen for new interactions between all Ig superfamily proteins, including antibodies and T-cell receptors, encoded by the human genome. This was a remarkable study recently published in Cell, with enormous potential to catalyze research on the role of these proteins in mediating interactions between cells in different organ systems throughout the lifespan. It was great fun to mentor Woj when she was a student with me, and it has been a great pleasure to continue to follow her work since she left the lab and to share our passion, joint passion for the wonderful world of cell recognition in the developing brain. The title of Woj's presentation today is Mapping the Cell Surface Interactome, Unraveling Biological Processes and Driving Translational Research. Welcome back to UCLA, Woj.
Thank you, Larry. Thank you so much. Let me just share my screen here. Am I sharing?
Yeah.
Awesome. Let me just get my slides to work here. Sometimes there's a delay in. Okay. Well, I have to say it's a total honor. It's really exciting to be here, to be back where I did my graduate work. As Larry said, what I'm going to talk about today is cell surface receptor ligand biology. And for me, this is just one of the most fascinating areas of biology. And I actually vividly remember the day when I was an undergrad in immunology class, when my professor, Frank DeToma, taught us about VDJ recombination and then how that gives rise to this incredible specificity. And this was, pre-PowerPoint, back when I think it was a requirement for being a professor to have this superpower where you could have this ability to write on the chalkboard with this inhuman warp speed, right? And so many of you will probably remember that, sometimes if you were lucky, you would have a small classroom and there would just be two blackboards. And so, when the professor would get to the end of the second one, they have to go back and erase the first one, and you'd have like 60 seconds to quickly try to catch up on all the notes. And when Professor DeToma was talking about this mechanism, I was sitting there and I was completely fascinated. I mean, I stopped taking notes. I was just sitting there listening. It was like my mind was being blown, and I was looking around, at all the other students who are, furiously writing and looking up and down, and I literally just wanted to yell out to everyone, like, don't you hear what he's saying? I mean, that moment for me was just, it really set the course of my entire scientific career. And today I still have this very same fascination with understanding how these physical protein-protein interactions at the cell surface, these events that allow cells to recognize one another and their environment and then respond appropriately, how all these recognition events mediate important aspects of all biological processes, from the immune system and cancer cell survival and metastasis to nervous system formation, function and regeneration, to a variety of developmental processes, as well as other processes like metabolism and vascularization, as well as interactions with pathogens. And of course, cell surface and secreted proteins are often dysregulated in disease states. So they serve as useful biomarkers, and then due to their extracellular accessibility, they comprise the majority of FDA-approved drug targets that have been developed in recent years. And so now, to understand on a global level how cell surface interactions mediate all of these processes, I would argue that as a starting point, there are three things that we really want to know. First, we would want to know who's there, meaning knowledge of all the secreted and cell surface proteins present in each cell type in the body, and then ideally what's expressed in each cell type at any given time in whatever context a cell finds itself. So what we might call a dynamic atlas. And now with all the amazing advances in single cell profiling, this is something that we actually can do and is being done both in individual labs and then through large initiatives like the CZI. And then the second thing I would argue we want to know is who can talk with whom, meaning which of these proteins physically interact as receptor ligand pairs. So a cell surface interactome map of all the interactions that can happen at the cell surface. And this is something that we're also making much progress in doing. And then lastly, I'd argue that we want to know what's the result of that conversation. Does it elicit attraction or repulsion, activation or inhibition, or whatever process you're interested in. And now here's where we can all see that it just got a lot more complicated, because in many, or perhaps most cases, the functional consequences of receptor ligand interactions are context dependent. So, we can't just take a receptor ligand pair and say, oh, this always causes activation of immune cells, or this always results in synaptic strengthening of neurons. And so to investigate the result of the conversation, we really need to study each interaction within its specific system, in vivo, or in cases where one exists in a functional cell assay. And these studies are often more time consuming and arduous, but many biological processes can now be studied on a scale that years ago was unimaginable. And I think we'll continue to see much progress in these areas. And so today I'm going to tell you about our efforts to get at the second step by global mapping of human cell surface interactions. So if we want to map the entire cell surface interactome, then what are we really talking about? Well, the human genome encodes about 2,000 single transmembrane cell surface proteins, about 1,700 with 2 or more transmembranes, and then about another 2,000 secreted proteins. So testing every pairwise combination of these proteins for interactions, isn't trivial. And if we focused on just the single transmembrane and the secreted proteins, then if we want to test every pairwise combination of the single transmembranes in a matrix, that's going to be 4 million combinations. And then if we also want to test every pairwise combination of single transmembranes and secreted proteins in a matrix, then that's another 4 million. So here, just with single transmembranes and secreted proteins, we're talking about 8 million interactions that need to be screened. So historically, the problem with doing this for cell surface protein interactions was that these proteins are not amenable to the standard protein-protein interaction screening methods like yeast-2 hybrid, which tests proteins for binding inside the cell. So as Larry mentioned, until about 15 years ago, cell surface interactions needed to be tested individually, or in small numbers using basic biochemistry techniques like immunoprecipitation or, purifying proteins and putting them on cells to look for binding, or using purified proteins in SPR experiments. And so I'm just going to go through a little bit of the history that Larry just told you about to explain and help you understand how and why the first assay was developed that allows cell surface receptor ligand screening. And I should just point out that variations on this assay, as well as different assays, have also subsequently been developed. And so as Larry said, just to put this into a historical perspective with some pictures on the screen, we need to go back to 2000 when Larry's lab discovered that this Dscam gene in flies encodes, as he said, 38,000 proteins, and these include 19,000 different extracellular domains. And so, when you're a lab like Larry's studying this problem, this, seemingly unfathomable problem of how from the limited number of cell surface proteins encoded in the genome, you can have enough cell surface recognition to allow millions of neurons to be able to recognize one another and then figure out, if you will, with whom they're meant to form synaptic connections during development. And of course, in humans, we're talking about billions of neurons forming many, many trillions of synaptic connections. When you're a lab that's studying this and you discover a gene that gives rise to 19,000 cell surface proteins with different extracellular domains, then, that certainly gets your attention. And so, as Larry said, the lab had done some genetics, and most of the Dscam phenotypes were pretty messy, so they weren't really giving us sufficient insight into how Dscam functions, or even whether this diversity of 19,000 extracellular domains is even required. And so, we decided to study the binding properties of Dscam proteins, and based on their similarity with other cell surface proteins, we predicted that they might engage in homophilic binding, meaning that Dscam interacts with Dscam on an opposing cell. And, as Larry mentioned, the problem was that that project that we wanted to do would have required making and purifying hundreds of different Dscam proteins to test binding between thousands of pairs. And I was the grad student doing this project, so the idea of purifying hundreds of proteins was not particularly appealing. And so what we really needed was an assay that would essentially bring the power of yeast-2 hybrid to the cell surface. And so in an attempt to do this, we developed an ELISA-based binding assay for screening cell surface interactions. And this assay just takes advantage of the fact that when cell surface proteins interact, it's the extracellular domains, right, that physically interact. And so when we developed this assay, we just did what's standard practice, and we made recombinant proteins where we fused the extracellular domain to both FC and alkaline phosphate tags. Both of these form dimers, and then we produce these proteins by transient transfection, and then, because they don't have a transmembrane domain, they get secreted out into the culture media. And then here's where we did something different. Instead of purifying these proteins, we simply mixed the FC and AP culture media in an ELISA plate that's been coated with an anti-AP antibody. And the two antibody binding sites can each bind to one dimer. So we end up with a tetramer. And if anyone's interested in why this is really important, we can talk about that later. And then we just do the same thing with an FC antibody. And of course, this HRP here allows us to detect binding between these proteins. So after incubation and washing, if this interaction occurs, then when we add the substrate, the well turns blue. As Larry said, we use this assay to screen thousands of combinations of Dscam proteins for binding to one another. And in the end, we found that each Dscam protein engages in highly specific homophilic binding to itself, but except in rare cases, no binding to different Dscam isoforms. And the way this works is that Dscam has 3 variable domains, And for each of these, there are many variants. And alternative splicing can shuffle these variants into any combination. So that's how we get 19,000 extracellular domains. And what we found in collaboration with David Eisenberg and Mike Swaya down there at UCLA, and along with David Baker at the University of Washington, is that each of these 3 variable domains binds to itself, because basically each one of these domains fits together. And so here, this would be Dscam on one cell interacting with Dscam on another cell. And then this isoform here, which has a different red domain, that one also binds to itself, but these 2 isoforms don't bind to one another because a mismatch at even one of the domains then prevents them from being able to fit together. And then through a large number of single cell profiling experiments and genetic analyses that were done by Daisuke Hattori and Satoru Muir in Larry's lab and in collaboration with other labs, we found that this homophilic binding specificity allows neurons to recognize self and mediates this process called self-avoidance, which is essential for patterning neurons in both the peripheral and the central nervous system. And so the way that this Dscam story unfolded really highlighted the power of biochemistry to drive our understanding of biological processes. And we reasoned that this ELISA-based platform could be employed to study a biological system using that approach that I talked about earlier, where we first identify who's there and then who can interact with whom, and then finally, what's the result of these conversations. And so when I started my lab at UC Berkeley as a Bose fellow, we used the mouse retina as a system to employ this approach, and where we could investigate cell surface interactions that mediate targeting of different subtypes of neurons that target to different layers in this region here. And for the who's there part of the project, we collaborated at Harvard because Jeremy Kay, who at the time was a postdoc in the lab, had profiled gene expression in 13 different subtypes of these neurons. And so we used Jeremy's data to identify cell surface and secreted proteins that are differentially expressed in different subtypes of neurons, because we reasoned that it would be these differentially expressed proteins that would be good candidates for mediating recognition events between neurons. And then we performed a binding screen with these candidates. And as Larry mentioned, we found about 50 new receptor ligand interactions. And we followed up on some of these binding pairs, both in vivo and then using primary retinal neuron culture assays. And we found a receptor ligand pair that's expressed in different neurons that target to alternating layers. And this interaction mediates repulsion of these neurons. So unveiling a repulsive model for this targeting specificity. And this mechanism is now being studied in vivo in mice using genetics by Jeremy Kay, who I should mention is now an assistant professor at Duke. And so all of this showed us that we could use this approach of, first figuring out who's there and then who talks with whom and what's the result of that conversation to learn something about a biological process. But these studies were really far removed from investigating cell surface recognition on a global level. They're really far from generating a large interactome that would be directly translatable to humans across many biological systems and multiple disease states. And it was really clear that this wasn't the sort of thing that we could get sufficient funding to do in a small academic lab. And then right around that time, Sean Parker, who many of you know as the first president of Facebook, and those of us who are a little bit older in the room probably remember as the founder of Napster, when for a few years we all, illegally downloaded music from the internet. Sean Parker provided a multi-million-dollar gift to Chris Garcia at Stanford to fund a human cell surface interactome initiative. And I had the opportunity to go there and direct this project. And as Larry said, the project was performed in collaboration with Caltech, as well as with GNF, which is one of Novartis's research institutes that's down in La Jolla. And at Caltech, the work was done in collaboration with Kaizen and Joost Vielmetter, who's the director of the Caltech Protein Expression Center and Robotics Facility. And so for phase 1 of the project, we selected 564 single transmembrane, GPI linked, and secreted proteins that belonged to the immunoglobulin superfamily and the tumor necrosis factor superfamily, and then along with a select group of additional proteins we were interested in. And we selected these families of proteins because they're known to be involved in all of these processes that I talked about earlier. And then we performed a binding screen, testing every pairwise combination of these proteins. And to do this, we started by curating all of the cell surface and secreted proteins in the human genome. And then we selected the ones that had immunoglobulin domains or were members of the TNF superfamily. And then we had these genes synthesized and subcloned into expression vectors to make these 2 different versions of proteins we need for the binding assay. And you might notice that this is a slight variation on the ELISA assay I showed you before, but it's conceptually the same. And then GNF made all the proteins using their incredible protein production robot. And then while all of that was happening, we optimized the assay down to a 384 well plate, and we developed a high throughput automated platform on the robots down at Caltech. And then, we performed the screen. And then, of course, as with any screen, there are going to be false negatives. And so to try to recoup some of these, we used our screen data in combination with phylogenetic homology analysis that we used to predict additional interactions that we didn't observe in the screen. So I should mention that while this assay has an extremely low false positive rate, we do see a significant number of false negatives. And then we validated these interactions, both ones that we observed in the screen as well as some of those that we predicted, and we validated them using surface plasmon resonance, or SPR, and cell binding experiments. And this whole part of the project here took a year, and then this actual screen itself took only 8 weeks. And as you can see here, a lot of 384 well plates. And we found 426 receptor ligand interactions, and of these, 81 were previously known and 345 were previously unknown. And partners for 254 of the proteins that we included in the screen. And so proteins that we didn't find binding partners for may have binding partners that we didn't include in the screen, or they may require co-receptors, or they may be false negatives because their binding fell outside the dynamic range of the assay, or it's possible, that some of them were misfolded. And so these 426 interactions form a complex network. And with the exception of 18 interactions, all of the interactions form a highly interconnected network. And this highly interconnected network is consistent with what Genentech found in a similar IG superfamily screen that they also published this past summer. And now, these highly interconnected networks are really similar to intracellular networks that we're all familiar with seeing. And I think it's interesting because highly connected intracellular maps have been evolving and growing for several decades. And we're all comfortable with the fact that, largely every protein inside the cell is known to have multiple binding partners. But, the cell surface mapping field has lagged behind. And I think with that, our understanding and appreciation of the fact that just like proteins inside the cell surface and secreted proteins each have many binding partners. And, it's hard to know how many binding partners when, only a portion of the cell surface interactome has been mapped to date. But just based on what we've done so far, I would estimate that on average, this is just me, it's likely going to be the case that each cell surface protein has about 10 or more binding partners. And, this is really different from how we've historically thought about cell surface interactions, because before these large scale cell surface interactome mapping efforts, so many fewer receptor ligand interactions were known, and it was just generally sort of believed that it was more the exception rather than the rule that cell surface proteins have many binding partners. And I remember when I was a student, in cases where many binding partners were found for a particular cell surface protein, people often referred to these as quote unquote promiscuous. And the implication there was that these many binding partners were more of an artifact, that they weren't actually functionally or biologically relevant. And I remember one of the faculty members in BC, although embarrassingly, I don't remember who it was, although perhaps someone who's here today remembers, or perhaps that person is here today. But I just distinctly remember hearing him say, there's no such thing as promiscuous binding. And, what I understood him to be saying was that bona fide protein-protein interactions, so, meaning proteins that exhibit physiologically relevant binding kinetics, that these undoubtedly have some cellular function, and I'm actually a big believer in the fact that, if this is not entirely true, it's at least largely the case. And, I think when we now think about it in hindsight, perhaps it makes complete sense that just like intracellularly, this has to be the case for the cell surface in order for such a limited number of cell surface unsecreted proteins to mediate the vast complexity of cellular recognition events and responses that are required to carry out this multitude of processes, both within and across all the different biological systems in the body. And consistent with this, the new interactions and the multiple binding partners we observe in the network involve proteins expressed in many different cell types and tissues that are involved in multiple biological systems and processes. And we found interactions between proteins both within a biological system and across systems. We also found binding partners for really well-studied receptors, for instance, the IL-6 receptor. And then we found binding partners for orphans, meaning receptors or ligands that prior to the screen had no known binding partners. And these proteins are known to be involved in multiple cancers and various disease states and disorders, pathogenesis, and many of them serve as important biomarkers or they're targets of approved drugs. And now we had decided from the very beginning of the project that once we had the screen data, we were going to select a large handful of varied interactions that we thought were potentially interesting, and then validate that they are indeed protein-protein interactions using SPR and cell binding assays. And then we would just publish all of the data to get it out there so that people studying the many different biological systems could start investigating the function of all these interactions in vivo. And so, if you're not familiar with how SPR works, I'll just quickly tell you that you capture a ligand on a sensor chip, and then you inject its binding partner, which is called an analyte. And then as the analyte flows by, it binds to the ligand. And then when the injection is stopped, the analyte dissociates over time. And then this binding and then the loss of the binding is measured optically. And so these SPR curves here with twofold decreasing concentrations of analyte, they show the association or the binding and then the dissociation. And you can see here that there's a concentration dependent binding. And when we do these SPR experiments, we always perform a negative control with a non-binding analyte. And then we also perform a positive control for this negative control analyte, just to ensure that the negative control analyte is functional for binding. And then for the cell binding assays, we use a purified ligand that's tetramerized with a fluorescently labeled streptavidin or SA here. And we incubate increasing concentrations of this ligand with transfected cells expressing the cognate receptor or a control receptor. And then we just monitor binding using flow cytometry. And so when we finished the screen, we had this huge list of hundreds of new interactions. And now we had this quote unquote problem of, figuring out which interactions to select for validation. And in the end, we selected the interactions for validation using 4 different pathways. And the first pathway that we followed was to look at interactions between multiple members of 2 different subfamilies, because subfamily interactions often regulate complex cellular interactions within a system, and they're often modulators of disease states. So when we see interactions between multiple members of 2 different subfamilies, this gives us some confidence that these are likely to be true physiologically relevant protein-protein interactions. And then for proteins that don't share any significant amino acid sequence homology and they aren't part of a subfamily, the next pathway that we investigated was function. Because if 2 interacting proteins are known to play a role in the same mechanism or pathway, then that gives us some confidence that these interactions may have physiologically relevant binding properties. For example, we found 2 myelination proteins, and these 2 myelination proteins are not phylogenetically related to one another, but both of them interacted with a subfamily of proteins that's expressed on leukocytes. So, we thought this was interesting because these interactions may play a role in inhibition of neuronal regeneration following injury. And similarly, we looked at proteins with shared expression, either because the proteins are known to be expressed in the same tissue, or they're expressed in cells that can reasonably be expected to encounter one another, such as immune cells and vascular endothelial cells. And then lastly, we looked at things that we knew have therapeutic relevance, either because they're targets of FDA-approved drugs or they're targets of intense drug research. And of course, in many cases, interactions fall into more than one of these categories. And so what I'm going to do is just give you a taste of the interactions that we followed up on using these different pathways. And to select subfamilies of proteins for validation, we used the phylogenetic homology analysis. And this was really important because, as you can see in this subfamily here, phylogenetic homology analysis is often required just to identify subfamily members, because quite frankly, they often just have different names that don't readily identify them as belonging to a subfamily. They aren't, sort of all conveniently named, notch 1, notch 2, notch 3. And as I mentioned before, this phylogenetic homology analysis allowed us to predict and test additional interactions that we didn't see in the screen as a way of recouping these interactions that were missed. And we were interested in this interaction between DCC and insulin-like growth factor binding protein like 1. And as its name implies, DCC was discovered in a colorectal cancer study. This was back in 1990. And since then, many well-characterized functions of DCC have been elucidated in the nervous system, but its role in colon cancer is not really well understood. There's some research that suggests it may be functioning as a tumor suppressor. And now we're interested in this interaction. We thought it was potentially interesting because both insulin and insulin-like growth factor binding proteins are often dysregulated in colon cancer, and they promote cancer progression. So we validated this interaction using SPR, but in our screen, we didn't see binding of DCC to this other insulin-like growth factor binding protein, which shares the highest amino acid sequence homology with this L1. So we predicted that this interaction may have been missed. And then when we tested it by SPR, we found that indeed this was the case. And then in another example with this same subfamily, we observed binding of 2 of these members here to ICAM5. And ICAM5 is a protein that's expressed exclusively in the brain. It functions in synapse formation, stabilization, and refinement. And then in addition, the extracellular domain of ICAM5 can be cleaved and has been shown to exhibit immunosuppressive functions through cytokine regulation. So it's believed it may play important roles in regulation of brain inflammation. And so we validated both of these interactions, as well as the 3 other subfamily members, which again, we tested based on our homology analysis. And in total, we predicted 34 interactions between various proteins based on homology, and we observed binding for 33 of them. So this demonstrated that this homology-based prediction is a useful tool to fill in the gaps for interactions that were missed in the screen. And so these here are all examples where we see interactions between multiple members of subfamilies, but it's often the case that proteins will bind with very high specificity to only one member within a subfamily. And we observed an example of this with the killer cell immunoglobulin-like receptors, which are called KIRs. And these are a highly polymorphic family of proteins that are expressed in natural killer cells in the immune system. The way it works is that NK cells initially express a stochastic combination of KIRs, and then this expression is refined during development to tune the cell's killing response threshold. And this ensures optimal self versus non-self discrimination. And the KIRs fall into 2 different categories: those that have these ITIM motifs in their cytoplasmic domain, which are inhibitory, and then those that have ITAM motifs, which are activating. And in the screen, we observed binding of the poliovirus receptor to only one KIR, this one here, 2D-L5. And here you can see this binding specificity in the raw OD ELISA data. And I should mention we also validated this interaction both by SPR and cell surface binding. And now the reason why we selected this highly specific interaction for validation is because this particular KIR is associated with increased virus susceptibility and reduced antiviral response to therapy. So this interaction may have implications for the role of this particular KIR in immunity. And so moving on from subfamilies, the next interaction I'll tell you about is an interaction that we selected based on shared expression. We thought this interaction between TIE1 and the high affinity nerve growth factor receptor TrkA was interesting, not only actually due to shared expression, but because both of these proteins, so both the ligand and the receptor are receptor tyrosine kinases. And TIE1 is expressed in vascular endothelial cells where it functions in this angiopoietin TIE2 pathway to inhibit angiogenesis. And in mice, loss of TIE1 in endothelial cells reduces primary tumor progression and it limits metastasis. And TrkA has many well-studied functions in the nervous system, but it's also expressed in vascular endothelial cells and in immune cells, where in both of these cell types, its function is not well understood. And so these 2 proteins are not only expressed in the same tissue, but then they're also expressed in cells that encounter one another. And so following our cell surface binding validation and our SPR validation, we decided to investigate this interaction biochemically a little further. Since TrkA is the high affinity nerve growth factor receptor, or NGF, we were curious whether TrkA can bind both NGF and TIE1 simultaneously. And so to test this, we just pre-incubated TrkA with NGF, and then we examined binding of TrkA alone in the absence of NGF to TrkA when it's bound to NGF. And when NGF was bound to TrkA, we saw this increase in resonance units that's proportional to the size of the NGF dimer, demonstrating that TrkA can bind both NGF and TIE1 simultaneously. So, whether in the presence or absence of NGF, TrkA interaction with TIE1 may play a role in angiogenesis, and or some other biological process. And then the other interaction that I'll tell you about that we investigated based on shared expression is a new receptor we found for leptin called ISLR2. It's also referred to as links. And this protein also functions in nervous system development and for brain connectivity. And it's also actually a receptor for vasorin. And now, as you likely know, leptin is a hormone that's released from fat cells in adipose tissue, and then it travels to the hypothalamus where it binds to its receptor, which is called LEPR. And this regulates both appetite by inhibiting hunger and increases energy expenditure. And you know, leptin also plays roles in other processes, but its role in the regulation of fat stores is its most highly studied function. The leptin mutant was a spontaneous mutation that arose at Jackson Lab like back in 1949. And these leptin mutant mice, they eat excessively to the point of obesity. They often reach a weight that's 3 times wild type. And then it wasn't until about 50 years later that the leptin receptor was found. And the leptin receptor mutant mice also exhibit this obesity phenotype. So, you know, as you might imagine, the leptin signaling pathway is a major target of the billion-dollar anti-obesity drug market. But the problem is that these mouse leptin and the leptin receptor obesity phenotypes are not phenocopied in humans. So it's believed that there are other players in the leptin signaling pathway in humans. And so it's for this reason that we thought a new leptin receptor is potentially interesting. And so after validating this interaction at the cell surface and by SPR, as we did on the previous slide I told you about with TRK and NGF, we tested whether leptin can bind both simultaneously to the LEP receptor and to ISLR2. And here you can see a complete loss of binding of leptin to ISLR2 when leptin is pre-incubated with the LEPR receptor. And on the right, you can see the loss of binding when we pre-incubated leptin with increasing concentrations of LEPR. And then lastly, we looked at interactions involving proteins that we know have relevance, either because they're signaling competent, such as these examples I'm showing you here, or they're targets of FDA-approved drugs, or targets of intense research. And here I'm only showing a subset of known drug targets that we found new binding partners for. And so knowledge of new binding partners for drug targets is important because these interactions may be leading to unfavorable off-target effects of drugs. For example, PD-1 is a negative regulator of T cell function, and its ligands PD-L1 as well as PD-L2 are expressed on tumor cells. And this interaction between PD-1 and PD-L1 inhibits immune response. There are several FDA-approved antibody drugs against PD-1 and PD-L1 that block their interaction. And these drugs can induce long-term remission for some lethal metastatic cancers, but these antibody drugs also can elicit serious immune-related adverse reactions. So that limits their clinical application. And in our screen, we observed binding of PD-1 to an additional previously unknown ligand called CCAM1, which is a member of the carcinoembryonic antigen subfamily, or CCAMs, of which there are 12 members. And the CCAMs are expressed in blood, bone marrow, and lymphoid tissue. So they're in the right place to be interacting with PD-1. And they play roles in cancer growth and invasion, and they're used as tumor markers for several different cancers. And now we also found that PD-L2 interacts with another member of the CCAM family, which prior to our screen was an orphan. So the fact that both PD-1 and PD-L2 interact with CCAMs, again, here's a case where this gives us some confidence that these are bona fide protein-protein interactions and therefore functioning in vivo. And now, because PD-1 antibody drugs can elicit serious adverse reactions, it's going to be important to investigate whether these PD-1 antibody drugs, which inhibit interaction with PD-L1, whether or not these drugs also inhibit PD-1 interaction with CEACAM1, right? Because if so, this would suggest that development of antibodies that specifically block interaction with PD-L1 could have more favorable clinical results. And so in this way, Interactome data can provide information not only for possible new drug targets, but it can also suggest current drug targets for which more highly specific drugs could have more favorable clinical results. And we think that Interactome data can be used as a pipeline for drug discovery. It can be used to identify novel receptor ligand interactions that involve proteins that are known to be dysregulated in disease states. We can then perform drug screening, for instance, using antibody libraries to identify drugs that block these interactions, whether they function as agonists or antagonists. And then these antibodies can be tested in mouse disease models to pharmacologically probe the effect of these candidate drugs, and of course, with the ultimate goal of developing therapeutics for various disease states. And so at this point, I just want to step back and look at the big picture. Because while we screened almost 320,000 interactions and found hundreds of new receptor ligand binding pairs, we've really only just gotten started on the road to cell surface interactome mapping. I told you at the beginning of the talk that if we want to screen all single transmembrane proteins and all single transmembrane with secreted proteins together, that's 8 million interactions. And these numbers are not even yet considering multi-transmembrane proteins. And then, in addition, they're not considering, proteins that require co-receptors or are mediated by cis heterodimers. We won't see these interactions in these types of screens because these screening platforms are only testing binary interactions. And I view co-receptors and complexes as sort of the necessary next step that we're going to need to address in developing screening platforms so that we can then layer these interactions involving complexes on top of all the binary data that we're collecting. And so in these recent IG superfamily and TNF superfamily interactome studies, both ours, and Genentech's, we've made headway toward mapping the cell surface interactome, but there's still much, much more to do. And these automated high throughput platforms, both the one we developed and the one that Genentech developed, they allowed us to screen many more interactions than was previously possible, but all assays have their limits. And as far as this ELISA platform goes, I think we're really approaching the limit of what makes sense to try to do with the throughput and the methodology for doing this type of screen. And I think that larger interactome studies in the future, ones that can screen both binary interactions as well as interactions with complexes, that's going to require the development of assays that take advantage of multiplexing. For instance, bioplex beads, which come in 500 different fluorescent colors. Each of these beads could each be coated with a different receptor, and then all the beads could be mixed together and then incubated with a fluorescently labeled ligand. And then the ligand receptor binding can be detected using an instrument that, just like a flow cytometer, flows beads single file past two lasers, one that detects the bead color, so that would tell you the receptor, and one that detects the ligand. And this experiment could also be conducted using beads with a single receptor and then a library of barcoded ligands, which could then be identified using MiSeq or some other sequencer. And then in addition to these methods, and of course there are others, there are additional strategies that are possible that would allow both the receptors and the ligands to be multiplexed in the same experiment, as well as cell-based assays that can be used both for single and for multi-transmembrane proteins. And, with all of these emerging new technologies and the platforms that we can build using them, I think that now we are really on the precipice of moving into a whole new era of interactome mapping that will drive our understanding of many biological mechanisms and processes, and along with that, provide valuable new therapeutic targets. And so my goal today was to tell you about the current state of cell surface interactome mapping and where I think we can go from here. And then to share with you how we think about digging into these interactome data in terms of using different pathways to find interactions that are potentially interesting, and to provide you with some examples of what we found and how we think about these interactions, how we think they may be functioning in vivo, and why we think that some of them may have therapeutic relevance. And then to suggest a strategy whereby interactome data can be used as a pipeline to drive translational research. So you know what, I think I will stop here and answer any questions if there are any. And if there aren't, I'd actually love to hear any thoughts and ideas that you may have, about what we as a community can do with all of these data to really push the boundaries of our current biological understanding. But before I do that, please let me hold your attention for one more slide while I acknowledge the many people whose work I just talked about. All of the Dscam studies I talked about were done in Larry Zapersky's lab, along with a variety of other collaborators. Here are the individuals who did the biochemistry, the structural biology, microarray expression, as well as genetics. The retinal studies I told you about were done at UC Berkeley with these members in my lab, and along with multiple collaborators that we had the tremendous fortune to work with. And the Human Cell Surface Interactome Initiative, as I said, was funded by a very generous gift by Sean Parker to Chris Garcia at Stanford. We had a wonderful team of people at Stanford, also a wonderful team of people down at Caltech. This was just an absolutely phenomenally fun project to do, and it would not have been possible without all of the resources and support and help at GNF. So thank you. I'm so glad that you came, and I'm happy to answer questions if there are any.
Great. Thanks, Woj. So I just want to encourage people, if you have questions, to go on to the Q&A feature and post your questions, and then I will select some of them and also ask my own questions as well. But we have a bunch of questions. Let me just start with a quick question about, sort of a simple question about discriminating between cis and trans interactions.
Yeah, that's a really good point. So I think what Larry's asking is, in these, some proteins are interacting in cis within the cell membrane. And when we do these types of screening platforms, presumably some of the interactions that we find will be cis interactions. And so I think the SPR and the cell surface binding experiments, when we see interactions in those assays, that really helps us to have confidence that these interactions are happening in trans. But we definitely, I assume we are seeing interactions in cis as well.
Great. I asked that question, and then as I was scrolling down, I noticed that exactly the same question was asked by your graduate student collaborator, Daisuke Hattori. So great, quite a happenstance. Anyway, let me go to another question here. I'm going to read it to you. Do you prioritize interactions with high affinity, low KD? This might be important for soluble, soluble, diffusible proteins binding to surface proteins. Maybe not so much for juxtacrine interactions between two surface proteins. Yeah,
that's a really great question. This is actually, so cell surface proteins, I don't know it's John. Hi, John. So as you know, cell surface proteins often have, what we would consider to be very low binding affinities, often in the tens of micromolar range. And so we really only see these interactions in our screening platform because we're clustering the proteins. We wouldn't see these interactions if we were using monomeric proteins because they're just very difficult to see using biochemistry techniques. But these interactions are also very physiologically relevant. When we choose the ones for validation, we don't yet know or have any idea what their binding affinity is going to be. But this is actually the reason, and I didn't mention this detail, of why we do our preliminary SPR using dimers, because we're really just looking for concentration dependent binding to convince ourselves that the proteins have physiologically relevant binding properties. We don't start with monomers because then, we would miss some of these interactions and then we would go back and test them as dimers.
So a somewhat related question from Owen Whitty deals with, I'll just read the question, but I think you might be able to expand on this a little bit, is how does your screen deal with low affinity interactions that depend on avidity enhancement for function? And maybe perhaps comment on the relationship between the binding that you see and KDs.
Well, I can see here Owen said, Woj just answered my question, but maybe I'll just tack onto that one more sentence, which is, the clustering forces that avidity enhancement. So the avidity is inherent in the assay itself.
Yes, but what's the range? So things that you pick up and then you look at for affinity by KDs, what sort of the range do you see? And do we know that those KDs are, that the protein interactions that have, let's say, low KDs or high KDs, sorry, are nonetheless biologically relevant?
Yeah, so some known interactions that have KDs around 50 micromolar, we can see in these assays. So some immune cell interactions, with very important functional relevance that have KDs that are, I always get it backwards to low affinity, high KD. We can see those interactions in the screen. I think the question here really is, do we miss the low affinity interactions? And then are we detecting interactions because of the clustering? Are we identifying, artifacts or protein interactions that are not, don't have physiologically relevant binding properties? And I can just tell you, for instance, for this screen, we selected, I think it was 67 proteins for validation, and 66 of them validated by SPR. So when I say that the screen has a very low false positive rate, that's what I mean when I'm saying that. And a lot of other labs have used this screening platform, and overwhelmingly, this is what everyone is finding.
So we have a question here which is asking whether you do have plans to look at transmembrane proteins, multiple transmembrane proteins. So presumably that it's really the ectodomain of multiple transmembrane proteins.
Yeah, well, I will just say we did sneak in a couple. I didn't mention we did sneak in a couple multi-transmembrane ones because this assay, if the multi-transmembrane just has one large N-terminal domain, then you can squeeze that one into this type of assay. When you have multiple extracellular regions that are coming together in form some three-dimensional structure, then it's just too much of a mess to try to fuse those together. So for multi-spanners, you really need to go into cells. There are some assays that are being developed. One that was published by Gavin Wright's lab at the Wellcome Trust Sanger Institute in the UK where they used CRISPR activating to generate a library of cells where each cell has a different cell surface receptor that's upregulated. And then you can go in, with a ligand and sort and then go in and sequence which of these CRISPR activating sequences is present in the cell. So you know which receptor was upregulated. These are not high throughput experiments. They're great experiments if you have a handful of orphan ligands that you really want to screen against a library. That works really great. But in terms of developing true high throughput, platforms, I think that's really difficult. IPI, the Institute for Protein Innovation, actually is getting a cell aggregation assay up and running, where you take two different cells that are fluorescently labeled differentially with a ligand and a receptor, and you mix them together. And this can be done in somewhat of a matrix format, but it's nowhere near the throughput of what the ELISA can do. But I think right now, if you want to do study these interactions on the surface of cell, we're just not at a place yet where you can do something at the throughput level that we really want to achieve.
So there's one question I'm going to rephrase here. I think this is what the individual is asking, which is really the question about producing proteins that are properly folded. And I wanted to get a sense of in your setup for producing proteins, what is the range of concentrations you end up getting, and are there sequences that are just protein sequences that are just very difficult to express, or is this expression system pretty robust and most things that you express are secreted and in some form that is in a relatively reasonable concentrations to do experiments?
Yeah, so for example, in this screen, we got 80, I think for one of either the AP or the AP, I think we got something like low 80% of proteins that were produced, and the high 80% for the FC. And now when I say produced, I mean they're detectable by Western. But this assay is actually so sensitive that you can detect micromolar interactions when the protein is present at picomolar concentrations. So sometimes we see interactions between proteins, and this is even with known interactions where we didn't see any protein on the Western. And then we put the proteins into the well and they're able to bind. And then in terms of like how many proteins are misfolded, I mean, I would expect that most proteins that are misfolded are not even getting out of the cell. But of course, some will be, and I think in those cases, most of those proteins are probably going to be sticky. We do, when we get the initial screen data, I think we had something like, I don't remember, 10 to 20 or so proteins that pretty much just bound to everything. So of course we throw those out. I expect that those are misfolded. When you see a protein that has, 5 or 6 interaction partners, you assume that probably, it's folded properly. The misfolded ones are resulting in false negatives or tons of false positives, which are really easy to throw out.
Great. So we have one last question from Dscam, and that question is, do you think computational methods, in particular, given that recent successes in the AI-based protein folding prediction that predict interacting surfaces partners, can perhaps narrow down the list of interactions that need to be tested?
Yeah, absolutely. I think this is exactly where we need to go. This is actually the problem with these Interactome data, is you have all of it and it's like, what do you do with it? You know, we want to look at various different things like Dscam, what you just suggested. You know, we want to look at expression. We also want to choose things that have, you know, high sequence homology between mouse and human, so we can do these types of, you know, therapeutic research. You know, we want to look at homology, but like right now, this is something that we have to do manually. I mean, we literally just make phylogenetic trees and say, okay, A interacts with B, and then we go and we look at the phylogenetic tree and we say, you know, what's in a subfamily with B, what's in a subfamily with A. You know, then we look through the literature to see, you know, what is, you know, for each protein, you know, what disease states is it associated with? You know, all of this is done manually. You know, so AI that could combine all of this, you know, and sort of spit out for us, you know, here's where the overlap in all of these different categories is. You know, this is exactly where we need to go. And once we start getting more interactome data, it's going to be, you know, almost useless because you can't even look through it all.
Great. So let's stop there. I want to thank everybody for participating, and particularly want to thank Woj for a fabulous talk with lots for us to think about in terms of the complexity of what's happening at the cell surface. Thanks a lot.
Thank you.
Take care.